So the third model of the specification of limbs according to the proximal distal axis is called reaction diffusion model. According to this model, the AER, you see the AER will divide the limb into three domains or three zones. The first zone just under the AER is called apical zone. The second zone is the active zone, and finally the frozen zone. The apical zone is basically the progress zone that you know, just under the AER. It's a region where FGF signals from the AER, okay, can act on it and can prevent the expression of specific ECM proteins like fibronectin. It's one extracellular matrix protein. Usually the fibronectin assembles the mesenchymal cells into nodules, into groups of cells, okay? But here in this apical zone, usually the presence of FGF in the AER will affect this very close zone under the AER called apical zone. This FGF will prevent the expression of fibronectin, so we can see separate cells in this zone. While in the zone called active zone, this zone is far away from the AER, okay, so it cannot be affected by the FGF. But this active zone is affected by another factor, another paracrine factor of the TGF-beta family, okay? So in this zone, TGF-beta will stimulate its own synthesis. You see, it's an autocrine loop for its own synthesis, so it will stimulate its own synthesis, but on the other hand, TGF-beta can also activate the expression of fibronectin in specific zones in order to group the cells into nodules, okay, or into groups of mesenchymal cells, okay? But if you look here in this active zone, we have nodules, but sometimes, so we can see between the nodules separate cells. So how it comes that we have separate cells? So this has been explained by the fact that TGF-beta will induce the expression of diffusible inhibitor. It's not known yet what is this diffusible inhibitor that will act to inhibit the expression of TGF-beta. So no TGF-beta, it means no fibronectin activation, and it means that these cells will stay separated and they cannot be assembled into nodules. That's why we have two populations of cells in the active zone, nodules and separate cells due to TGF-beta activation or not in these different parts of the zone. And finally, the frozen zone is made up by the freezing of the nodules, okay, the nodules that will become cartilage, okay, and then after ossification, this cartilage will give the bone. So this is called the reaction diffusion model, and you should know here that no single model of the three models I explained show the experimental phenomena or the experimental process about the generation of the proximal distal axis of the limb, but this process of the generation of the proximal distal axis, it's still under intensive investigation in order to understand the molecular mechanisms for limb formation according to this proximal distal axis. So in the limb bud, we talked about the AER, which is a main signaling center responsible for the formation and the growing of the limb according to the proximal distal axis. The main agent of the AER is the FGF8 that will induce all the time the expression of FGF10 in the progress zone. The second signaling center I want to talk about now, and it's responsible for the anterior posterior growth of the limb, is called the ZPA. The ZPA is the zone of polarizing activity, okay? So it's a region of the posterior mesoderm. So this is the posterior mesoderm, this is the anterior mesoderm. Okay, this is where the ZPA is usually located. So to understand better the function of the ZPA, this experiment has been done. So a ZPA is grafted from a limb bud, which is donor, from a donor limb bud, to be grafted into the anterior part of the mesoderm of a host limb bud. Okay, so that in the host, okay, limb bud, we have already the normal ZPA, normally located posteriorly, and we have the extra transplanted ZPA located anteriorly. So as a result of this experiment, we got this phenotype in chicken. Okay, so we got a duplicated digits. Okay, you see we have the stylopod, the zygopod, the otopod. So we have duplicated digits that will emerge, but they are oriented in a mirror image, okay, of the normal digits. Let's say here we have the normal digits, okay, and here we have the extra digits resulting from the transplantation of the ZPA. But these digits are arranged in a mirror, let's say mirror image configuration. So now, after discovering the ZPA, they looked at the main agent of the ZPA. What is the main active agent of the ZPA? Like, for example, we had FGF8 in the AER. So what is the main agent of the ZPA? So after making different in situ hybridization to look at the expression of the genes present in the ZPA and what are the genes that will co-localize with the ZPA and they might having a main function in the ZPA activity, finally they found the sonic hedgehog, okay, the sonic hedgehog gene. This sonic hedgehog is expressed at the level of protein in the ZPA, and it seems it co-localized with the ZPA location right at the posterior mesoderm. So to understand better about the function of the sonic hedgehog and how it acts as and if it acts as an active agent of the ZPA, they did the following experiments. So they took, in this case, fibroblastic cells from a chicken embryos, and they infected these cells by vectors, okay, a viral vector expressing the sonic hedgehog gene, okay. So these cells are induced to express sonic hedgehog. After some times in culture, they centrifuged the cells. So we have the pellet here and here the supernatant. So the pellet will contain the cells expressing sonic hedgehog. So they took the pellets, okay, and they implanted them in the anterior portion of a limb bud. It means just right here. These are the cell pellets that will secrete and express sonic hedgehog. So after some times, they let this limb bud develop, so they got this phenotype. As we can see, the resulting limb produced a mirror image digit. This is the normal development of the limb, and this is the mirror image duplication coming from the graft, okay? And as you can see, we obtained exactly the same phenotype as we grafted the whole ZPA, as I showed you right here in this experiment. Here we got exactly the same phenotype. So what does it mean? It means that the main active agent of the ZPA is sonic hedgehog. Since if we graft cells expressing only sonic hedgehog, it will mimic exactly the same phenotype obtained when we graft the whole ZPA. So as a conclusion, sonic hedgehog is an active agent of the ZPA. So how the sonic hedgehog functions during the formation of the digits, as we have seen that sonic hedgehog is the main agent of the ZPA. Here we have an early mouse hind limb. You see the mouse hind limb bud is formed by a mesenchyme which is not segregated into digits at the beginning. So in the ZPA zone, okay, right here, we can see two dots. The green dot okay, presents the progenitor cells for the digit number 4 that will be formed in the future, while the red dots presents the progenitors of the digit number 5, okay? So at the beginning, the progenitors of the digit 4 and 5 are present in the ZPA. It means in the region that will express sonic hedgehog, okay? This light green region expressing sonic hedgehog. So sometimes after, you can see that the cells forming the digit number 5, okay, will still present in the ZPA region. It means are still expressing sonic hedgehog in the ZPA, but the cells that will form the digit number 4 are no longer present in the ZPA, so they exit this zone, the zone of the ZPA. So when the digits will be formed, you see that the cells in the digit number 5 will have seen high levels of sonic hedgehog because they stayed longer in the ZPA zone, so in the zone of sonic hedgehog expression. So it means that they have seen high levels of sonic hedgehog for a longer time than the cells in the digit number 5. Okay? So in this figure in D, we can see it's a schematic showing the digits number 4 and 5. It's the zone of the digits number 4 and 5. You see in this zone, sonic hedgehog is expressed in an autocrine way, but also in a paracrine way. So sonic hedgehog can diffuse from this zone to the neighboring zone, the zone of the digit number 3. Okay, so the zone of the digit number 3 can also produce sonic hedgehog by autocrine way, but also it can receive it by paracrine way through its diffusion from this zone of the digit number 4 and 5. While the zone of the digit number 2, okay, can also, while the zone of the digit number 2 will receive only sonic hedgehog through paracrine diffusion, so this zone cannot produce sonic hedgehog in an autocrine way. But interestingly, you see that the digit number 1 is completely sonic hedgehog independent. So we don't know exactly what is the factor that can, which is responsible for its formation, but it's completely independent from sonic hedgehog concentration. So in the next slide, or before, I have to mention that the sonic hedgehog secreting cells, okay, the secreting cells in an autocrine way, form digits number 4 and 5, and they contribute to the specification of digits number 3 and 2 through the paracrine way. It means through the diffusion of sonic hedgehog. So here it's the same idea. We have the zone of the digit number 4 and 5. Okay, so this zone is called the descendants of sonic hedgehog expression. It means that these cells or these zones will express in an autocrine way sonic hedgehog through a paracrine way. Okay, the sonic hedgehog will diffuse. Okay, so digit number 2 has low sonic hedgehog concentration. Okay, since it only receives sonic hedgehog by paracrine diffusion, while the digit number 5, the digit number 4, and the digit number 3 can produce sonic hedgehog in autocrine way. Okay, 4 and 3 can receive it from the digit number 5 by a paracrine diffusion. Okay,